entitled Procedures for FDA Staff, In Vivo Bioavailability Bioequivalence Studies, Clinical, outlines procedures for FDA investigators who inspect domestic or international sites to ensure that the clinical portions of in vivo BE studies, including studies with pharmacokinetic, pharmacodynamic, or clinical endpoints submitted to CDER, are conducted in a manner that ensures subject safety and integrity and data integrity, and to document study conduct in accordance with applicable regulations. The CP is designed to ensure studies are conducted using the highest laboratory standards and in accordance with applicable regulations. The first presentation in this session will be by Dr. Xingfang Li, who will discuss the inspectional approach for the clinical portion of the in vivo BE studies as described in the compliance program. Next, Dr. Xikui Chen will discuss the inspectional approach for the BE studies with clinical endpoints. Finishing up the session, Dr. Paul Lihong Ye will cover procedures for handling reserved samples from relevant BA and MBE and the expectations regarding responsibilities of each party involved in the study pertaining to reserved samples. Now for some information about our presenters. Dr. Xingfang Li is a pharmacologist in the Division of Generic Drug Scientific Integrity within OSIS. Dr. Li started at FDA in 2010 as an FDA's Commissioner's Fellow in the Office of the Chief Scientific Officer, and then joined OSIS in 2012. And Dr. Li has coordinated, conducted, and reviewed FDA BIMO clinical and analytical inspections in support of NDAs, ANDAs, BLAs, biosimilars, and INDs. Dr. Xikui Chen is a pharmacologist also in the Division of Generic Drug Study Integrity within OSIS. He received his PhD in pharmaceutical sciences from the University of Oklahoma in 1996. Dr. Chen has conducted analytical inspections as well as participated in clinical inspections and evaluated inspectional observations since joining the Division of Scientific Investigation in 2007. Dr. Ye has joined FDA since 2014. He graduated from the University of Missouri and had postgraduate training at the Johns Hopkins University. He is interested in bioengineering, bioanalytical analysis, and biologics products review, inspection, and regulation. Please join me in welcoming all our presenters.
Thank you for your introduction. Welcome to attending the OSIS SBIA workshop. I'm a reviewer in the Office Study Integrity and Surveillance, Office of Translational Science, CDER, US FDA. My presentation today is titled Inspecting BAB Clinical Studies. This presentation will outline the basic elements to be examined during the clinical inspection for in vivo BAB studies. Before starting my presentation, I would like to read this disclaimer. The views and opinions presented here represent those of the speaker and should not be considered to represent advice or guidance on the behalf of the U.S. Food and Drug Administration. The compliance program 7348001 used to cover inspection of both clinical and bioanalytical components. After 2018 reversion 7348003, which I will describe the CP, is focused to auditing clinical component only, and others from OSIS will describe reserve samples and case studies. Here's the outline of my talk. Objective of the compliance program, the scope of the compliance program, documentation expectation, and the current compliance program. Then the summary. The objective of the implementation in vivo BAB Bioresearch Monitor Program to ensure the protection of the right, safety, welfare of the human subject participating in drug studies, to ensure the quality and integrity of the clinical, analytical, and statistical data from BAB studies, to ensure compliance with the applicable FDA regulation, and to identify significant deviations. 7348003 outlines the procedures for FDA investigates who inspects domestic or international site to ensure the clinical portion of in vivo BAB study are conducted in the manner to ensure subject safety and data integrity, and to document study conduct in accordance with applicable regulation. Clinical portion of the in vivo BAB study, there are several different types. Studies with the pharmacokinetic endpoints, study with the pharmacodynamic endpoints, study with the clinical endpoints. For this type of study, our colleague Dr. Xikui Chen will have a presentation today after this. The regulated industry and the CP 48003 is the clinical site, not the individual clinical investigator. Clinical site may include independent contract research organizations or exist as part of the pharmaceutical company or other institutions such as hospital or universities or individual doctor offices. Clinical and analytical portion of BAB study may be conducted at the same site or different site. BAB clinical inspection assignment can be issued by OSIS reviews based on the criteria of surveillance inspection, requests from CDER office who review drug applications, and the complainants and whistleblowers, self-identified or anonymously, from the public and private sectors. OSIS review may accompany ORA investigate during inspection as subject matter expert if needed. For the current process, OSIS issue inspection assignment memo to ORA. ORA investigate will conduct clinical BAB inspection. For routine BAB inspections, it usually takes 1 week of on-site inspection time. During the inspection, OSIS review provide on-site support to ORA investigate, including parts of the inspections as needed. To evaluate the overall quality of subject safety and data integrity, and 7348003, at a minimum, those 11 components will be examined during a BABE clinical inspection. This is not meaning to be an all-inclusive list of the components that may be covered during an inspection. The expectation for 8 of the 11 components have been explained very well in this CP. Dr. Ye will present the topic of the reserve samples based on the past inspection conducted by ORA Investigate. I will focus on the highlight 2 components, randomization and the blinding code. Now I would like to start talking about randomization schedule. Randomization schedule is used in the clinical trial to assign subjects randomly to one of the treatment regimens studied in the trial, usually generated during the study design phase to reduce any possible biases it may arise in the trials. Randomization schedule applies to both open label and blind clinical trial. Open label trial is a type of study in which both healthcare provider and the subject are aware of the drug or treatment being given. Blind clinical trial, a type of study in which subjects, single blind, and the subject and their providers, double blind, do not know which drug or treatment being given. This slide illustrates the process how to assign a treatment in an open label study. The site personnel follow the randomization schedule generated prior to the study initiation. The qualified subject return to site, and the site personnel will review the eligibility again. Then follow the randomization schedule to dispense the treatment. The subject and the personnel are aware of the treatment. If the randomization schedule is utilized during the study, the randomization schedule may be reviewed and could be collected during inspection. This is the example randomization schedule for the open label trial. You will see that there is the 4 columns: subject ID, sequence, period 1, period 2. So when qualified subject return to site, so they will assign sequence number, either 1 or 2. So 1 means TR, 2 is RT. So the subject 1 signed 2, that means period 1 reference, period 2 the test. For this open label study, the subject and personnel all will watch the drug the subject has received. This slide illustrates a double-blind trial. It differs from the open label in that subject and the site personnel does not know the treatment subject received. The process for this double-blind trial, the qualified subject will be assigned a randomization number based on randomization schedule generated prior to the study initiation. The randomization number is associated with a kit number, not exact treatment. The kit will be distributed to the subject based on the randomization schedule without revealing the treatment. here is a double-blind trial randomization schedule. So from the previous slides, we know the qualified subject come in, return to the site personnel will assign randomization number. For this schedule, you will see the randomization number in this column is called sequence number. So the qualified subject come in, the personnel assign 3296 for this subject. But the 3296, based on this randomization schedule, is associated with this key number. It's A1076. So this, the subject and the personnel don't know what the treatment the subject will receive. For the double-blind trial, FDA ORA investigator will compare the randomization schedule submitted to FDA with the randomization schedule maintained at the site. For the paper-based documents, we would like to know when the clinical site received the schedule, who has accessed it. For application software, we will request printout randomization schedule from the software. If any difference, request access to auto trail to trace those changes and evaluate the justification. If no access to auto trail, for example, the study is closed, or investigative requests the printout randomization schedule dated on the study initiation day and the study randomization schedule dated on the database log day, and evaluate the changes and the justification. I have mentioned in previous slides about the application software used during the clinical trial. So there is the interactive web response system, RWRS, and interactive voice response system. Other technologies that clinical research sites used to enroll subjects into the clinical trial, randomize subject, and manage study drug supplies. RVRS, RWRS can be accessed through telephone and web. It is more user-friendly and easy to access from anywhere in the world through the telephone and web. But there is another technology term. It's called interactive response technology. It's RRT. Basically, the RWRS, RVRS fall under the umbrella, the term, under this RRT term. Now I like to talk about blinding code. So blinding codes are used to tracking drug products, test, reference, placebo when applicable, given to each study subject without revealing the product identity for the subject or to the study personnel. There is different format, the blinding code. There is two-part tear-off, scratch-off label attached to the kit container. There are sealed code break envelopes. There are electronic version maintained in the software. For blinding code, we will determine accessing the blinding code, determine how and when blinding code was provided to the clinical site, document when the blinding code remaining at the clinical site throughout during the study, evaluate access to, handling of, storage of the blinding code during the conducted study and after study completion. And for breaking the blinding code, during inspection, FDA will perform the breaking the black code to verify the subject received the correct treatment. And during the clinical trial phase, if there is an emergency, so clinical site personnel can break the blinding code. We'll have a note to detail why you break the blinding code. As I mentioned in previous slides, there are several different types of blinding codes. This slide showing the one type is the scratch label. So this left panel shows intact blinding code. It's detached from products and place this sticker on the investigation of product dispensing record. So this will be in the subject research documentation, one of the. So when the ORA investigates, during inspection, they will scratch off this to reveal what is the real treatment. Then ORA investigator will initiate this. Here is another example, sealed envelope. Usually, this envelope will come with the drug shipment. This sealed envelope will always remain in the clinical site, in the cell, except there is an emergency situation. The site personnel will open this envelope to check the subject receive the treatment. However, after opening it, so the site personnel will detail the situation. The right side indicates the envelope inside. So it's a K number, and this is a real treatment. Here is another example. It's a printout blinding code from the RVRS, RWRS system. So this is from the software. This is the column is a K number, and this is the treatment. So in case emergency situation, the site personnel can unblind the blind code through the RVRS, RWRS system. But however, the site personnel will detail in the subject research documents why they unblind this specific subject. When auditing the blind study, we would like to ask those questions. Ensure the study was blinded per protocol when using RVRS, RWRS. Here is a summary for my talk. CP7348003 outlines the basic elements to be examined during the BABE clinical inspection. There are minimum 11 components. Documentation of study-related activities is critical during the conduct of clinical BABE study. I would like to thank Dr. Xikui Chen, Dr. Michael Scully, Dr. Kimberly Benson, Dr. Julie Chu, Dr. Doug Baum, and OSIS management for their great contribution to this presentation. Thank you for your attention. Let's have the following two challenge questions. Challenge question 1. Which of the following reports will be audited or reviewed during the clinical inspection for in vivo BABE studies? Inclusion, exclusion criteria of subjects, collecting, processing, and storage study samples, subject to bioanalysis, informed consent documents, randomization schedule, all of the above. I'll give 5 seconds. The answer is all of the above. The second question, challenge question, true or false? For closed studies, ORA investigate must unblind the blinding code to assure the study subject received the assigned treatment. That was reported to FDA in the study report. However, ORA investigators usually do not unblind ongoing studies if there is no safety concern. There is true or false, two answers. I give you 5 seconds as well. The answer for this challenge question is true. The reference related to the presentation are listed in the slides. I sincerely appreciate your attention, and that brings us to the end of my presentation. Thank you.
Hello everyone. The topic I'd like to talk about today is inspecting bioequivalence studies with clinical endpoints. Before starting my presentation, I would like to read this disclaimer. The views and opinions presented here represent those of the speaker and should not be considered to represent advice or guidance on behalf of the U.S. Food and Drug Administration. Learning objectives include the following. First, to understand what are clinical endpoint bioequivalence studies, followed by understand different product types for clinical endpoint bioequivalence studies and clinical endpoint assessments, and finally, understand inspectional findings of clinical endpoint bioequivalence studies and how OSIS evaluations of the findings affect data acceptance for the studies. Outline of the presentation comprises the following items. First, what are the clinical endpoint bioequivalence studies? And then, different product types and clinical endpoint assessments for clinical endpoint bioequivalence studies, followed by inspectional findings of clinical endpoint bioequivalence studies, and how OSIS evaluations of the findings affect data acceptance for the studies, and finally the challenge questions. A clinical endpoint bioequivalence study is defined as a comparative clinical study of two products intended to deliver the same active moiety at an equivalent rate and extent to the site of activity. This approach may be applied to the following three situations. First, dosage forms intended to deliver the active moiety locally. Second, dosage forms that are not intended to be absorbed. And third, drug products for which traditional pharmacokinetic studies are not feasible. Clinical endpoint bioequivalence studies are utilized when a pharmacokinetic study alone cannot reliably establish bioequivalence, or when locally acting products have minimal systemic absorption. OSIS uses the same bimocompliance program for clinical parts of biosimilar studies. This table lists examples of different product types and clinical endpoint assessments for clinical endpoint bioequivalence studies. The products for clinical endpoint bioequivalence studies include topicals, eye drops, nasal sprays, inhalers, dermal patch, vaginals, as well as suppository medications. The clinical endpoint assessments for clinical endpoint bioequivalence studies are evaluated based on desired outcome, clinician's evaluation, and the subject-reported diaries. For topical steroids, skin blanching was measured by chromometer. Inflammatory lesion or bacterial counts were analyzed for topical antibiotics. For non-steroidal anti-inflammatory drug, summary pain scores were read. For nasal spray, total nasal symptom scores were determined. Forced expiratory volume was measured for inhaler. For dermal patch, the adhesion, irritation, and sensitization were reviewed. Cytology and the total symptom scores were assessed for vaginal cream, and for suppository symptom summary scores were evaluated. Inspectional findings of clinical endpoint bioequivalence studies and OSIS evaluations of the findings for potential impact on acceptability of data are illustrated in the following three case studies. Those case studies are examples from the previous inspections or recent record reviews for clinical endpoint bioequivalence studies. Each case study discusses study design, objectionable findings, and OSIS evaluation. For clinical endpoint bioequivalence case study 1, the study design was a randomized, double-blind, placebo-controlled, parallel design, multiple-site bioequivalence study with clinical endpoints. Objectionable finding was that the clinical site did not retain reserved samples for the third shipment of products for the study. Based on OSIS evaluation, we recommended not to accept data from subjects dosed with kits from the third shipment of products. For clinical endpoint bioequivalence case study 2, the study design was a randomized, double-blind, multi-center, 3-arm, active and placebo-controlled parallel study to evaluate the bioequivalence with clinical endpoint. Objectionable findings was that the baseline data from 48% subjects enrolled for the study were reported to have the same inflammatory lesion counts, which are the sum of inflammatory papules and pustules on the subject's skin. Based on OSIS evaluation, we recommended that data from the study conducted at the clinical site are not reliable to support regulatory decision. For clinical endpoint bioequivalence case study 3, the study design was a double-blinded, randomized, placebo-controlled, parallel group treatment bioequivalence study with clinical endpoint. Objectionable findings were that the clinical site did not maintain a complete sealed blinding code, and the result samples were not retained at either the inspected clinical site or an independent third party. Based on OSIS evaluation, we recommended that the identity and accuracy of the study medication administered cannot be verified. In summary, the clinical site failed to meet requirements specified in bioresearch monitoring compliance program, may result in objectionable findings at inspection site, and may affect data acceptability from OSIS reviewer for clinical endpoint bioequivalence studies. I'd like to thank Dr. Xingfang Li, Dr. Michael Steady, Dr. Kimberly Benson, Dr. Julia Chiao, Dr. Cynthia Zhang, and OSIS management for their great contributions to the presentation. There are two challenge questions. For challenging question number 1, true or false? There is no documentation requirement for clinical endpoint bioequivalence studies. A. True. B. False. The correct answer is B. False. For challenging question number 2, under which of the following situations would a clinical endpoint bioequivalence study be conducted? A. When a pharmacokinetic study alone cannot reliably establish bioequivalence. B. With locally acting dosage forms. C. Dosage forms with minimal systemic absorptions. And D. All of the above. The correct answer is D. All of the above. The references related to the presentation are listed in the slide. Thank you for attending the presentation.
This session is overview of reserved samples. I am Li Hongye. I'm a chemist in the Office of Study Integrity and Surveillance in OTS, CDER, FDA. This presentation reflects the views of the author. It should not be construed to represent FDA's views or policies. In this session, we will review U.S. FDA regulations on reserve sample requirement for bioavailability and bioequivalence study, including in vivo BA/BE study in case number 1, in vivo BA/BE study conducted under a bio-IND in case number 2. Also, we will talk about good documentation practices during BA/BE study conduct, as in case number 3. Some background information. 21 CFR Part 320 tells us bioavailability and bioequivalence requirements. Specifically, in Section 320.38 is retention of bioavailability samples. Section 320.63, retention of bioequivalence samples. Regulations for reserve sample requirements is in 320.38, retention of bioavailability samples. Section A stated, the applicant of an application or supplemental application, or if bioavailability testing was performed under contract, the Contract Research Organization, the CRO, should retain an appropriately identified reserve sample of the drug product for which the applicant is seeking approval, of the test article, and of the reference standard used to perform an in vivo bioavailability study in accordance with and for the studies described in paragraph B of the section, that is representative of each sample of the test article and reference standard provided by the applicant for the testing. In 320.63 of retention of bioequivalence samples, the applicant of an abbreviated application or a supplemental application submitted under Section 505 of the FDIC Act, or if bioequivalence testing was performed under contract, the CRO should retain reserve samples of any test articles and reference standard. Regulations for reserve sample requirement for BA and BE study conducted under a bio-IND is in Section 320.31. Applicability of requirements regarding an investigational new drug application. A. Any person planning to conduct an in vivo bioavailability or bioequivalence study in humans shall submit an IND if 1. The test product contains a new chemical entity. 2. The study involves a radioactively labeled drug product. Or 3. The study involves a cytotoxic drug product. In Section 312.57, Record Keeping and Record Retention, Part D, a sponsor shall retain reserve samples of any test article and reference standard identified in and used in any of the bioequivalence or bioavailability studies. Section 312.52, Transfer of Obligations to a CRO. Section A, a sponsor may transfer responsibility for any or all of the obligations set forth in this part to a contract reserve organization. Let's take a look of case number 1. It is a BE study submitted under 505J to support an ANDA. The regulations is in section 320.38 and 320.63. To review, the contract reserve organization shall retain reserve samples of any test article and reference standard used in conducting an in vivo bioequivalence study. Each reserve sample shall be adequately identified so that the reserve sample can be positively identified as having come from the same sample as used in the specific bioavailability study. There are 4 key players in the IP handling. The sponsor prepared the IP shipment and shipped to packer, which set aside some reserves and then make the IP shipment to the clinical site. And there are some reserves for the clinical site as well as used for dosing. And there are some IPs are unused as shown here. And the clinical site also shipped some reserves to the third party, and the unused was kept in the clinical site, but the used IPs were shipped to the third party for storage. FDA conducted inspection of the clinical site. The outcome is objectionable findings were observed. Specifically, the reserve samples were not retained from all shipments provided for testing in the study. The clinical site indicated that the sponsor instructed them to retain reserve samples from shipments 1 and 2 only, but not from shipment 3. After study completion, the clinical site shipped the reserve samples, used and unused IPs to the third-party storage site. And representative reserve samples have to be retained from all shipments of drug products received by the testing site. Our evaluations are: because the clinical site did not retain reserve samples for reference and test product from shipment number 3, authenticity of the drug products used in part of the study that used drug products from that shipment could not be verified. The clinical site shipped the used and unused IPs from shipment 3 to a third-party storage site. The used and unused IPs from shipment 3 were retrieved from the third-party storage site and become available for review and verification during the remote record review. The used and unused IPs from shipment number 3 were verified and were used to mitigate the impact of not retaining reserve sample from shipment number 3. Let's take a look of case number 2. It is a pivotal in vivo BE study with PK endpoint submitted under 505(b)(2) to support an NDA application. The BE study had an associated bio-IND. The regulations are: a sponsor shall retain reserve samples of any test article and reference standard identified in and used in any of the BE or BA studies. A sponsor may transfer responsibility for any or all of the obligations to a CRO. What is bio-IND? Bio-INDs are investigational new drug applications submitted for bioavailability or bioequivalence studies under 21 CFR 20.31. The bio-IND is required by regulations in specific instances to ensure that proposed drug products are safe for use in human test subjects and do not expose them to undue risk. 21 CFR 320.31a states that any sponsor planning to conduct an in vivo BA or BE study in humans shall submit a bio-IND if 1. The study involves a radioactively labeled drug product, or 2. The study involves a cytotoxic drug. In case number 2, the BE study had an associated bio-IND. The regulations are a sponsor shall retain reserve samples of the test, article, and reference standard, and a sponsor may transfer responsibility for any or all of the obligations to a CRO. In case number 2, there are 3 key players: the sponsor, the clinical site, and the third party. Let's take a look at their roles individually. For the sponsor, they are responsible for retaining reserve samples of any test article and reference standard used in conducting an in vivo BE study. The sponsor may transfer the responsibility to a CRO. For the clinical site, it's dedicated by the sponsor to be responsible for selecting reserve samples of the IPs used in the study. For the third party, it's delegated by the sponsor to be responsible for retaining reserve samples of the IPs used in the study. Inspection outcomes. The clinical site selected the reserve samples and shipped them to the independent third-party storage facility. Evaluations. Per the study protocol, reserve retention samples of each study drug were maintained by the sponsor's designee. The sponsor transferred the responsibility of selecting reserve samples to the clinical sites and the responsibility of retaining reserve samples to the third-party storage facility. After completing the study, each participating clinical site shipped the retention samples to the third-party storage facility as specified by the sponsor using a carrier that maintained proper temperature storage conditions for the duration of the shipment. FDA investigators verified the reserve samples retrieved from the third-party storage facility during the inspection. This case met the regulatory requirements per 21 CFR 312.57 Part D. Therefore, there was no regulatory violation. Let's take a look at case number 3. It is an in vivo BA study with PK endpoint submitted under 505(b)(2) to support an NDA. The sponsor used a CRO to handle PK sample collection and processing at extended time points after dosing. After inspection, objectionable findings were observed. Specifically, documentation of the study subject's PK sample collection and/or processing at certain time points were missing or inconsistent. Evaluations. Without the adequate and accurate documentation, it was impossible to reconstruct the conditions those subject samples underwent under collection and processing. We concluded that some concentration data of the affected subject samples are not reliable based on the available information provided by the clinical sites and the CRO responsible for PK sample collection. Concluding remarks. Become familiar with the relevant US FDA regulations on reserved sample requirements for BA and BE studies, including in vivo BA/BE study and in vivo BA/BE study conducted under a bio-IND. Understand the importance of good documentation practices during BA/BE study conduct and potential impact on study data integrity. This presentation was assisted and supported by the following colleagues: Charles Boniface, Arindam Dasgupta, Gopal Biswas, Cynthia Zhang, and also OSIS management and colleagues.
All right, thank you for the informative presentations. We will now have our Q&A panel with all the session presenters. And if you haven't done so already, please take the time to enter your questions into the Q&A chat pod now. Our first question will go to Dr. Xingfang Li. Dr. Li, what would happen if there was an unintended unblinding of an ongoing study?
Thank you, Laurel. Can you hear me? Hello?
Yes.
So, yeah, thanks, Laurel, for your questions. I think that is a great questions. So I will repeat the question, what will happen if there was an intended unblind ongoing study? So if this protocol is designed to blinding study, this is an intended unblind. So I will like this clinical site to inform RRB and to inform sponsor to tell, you have an unintentional unblind this ongoing study that decided how to proceed to next. Should be, if the study design is unblind, should be immediately inform the sponsor and RRB and to take following actions. Thank you that's the answer.
All right, thank you so much for that response. Our next question is coming for Dr. Chen. Dr. Chen, what is the problem with case 2 study? The severity of the disease appears comparable among the patients, or do you mean that the data is manipulated?
Thank you, Nora, for that question. For that case study, because the inflammatory count that was used on the baseline and the effectiveness based on the after dosing, they recount the inflammatory numbers and to see the effectiveness. And 48% subjects enrolled, they have the same exactly the inflammatory count number is kind of questionable. So that's why we give the recommendation to the review divisions, because that's the critical number for the baseline in this clinical endpoint BE studies. Thank you.
All right, thank you for sharing that information. We'll go back now for a question for Dr. Li. What are the main purposes for maintaining sealed blinding codes at each clinical site?
Thank you, Laurel, for the great questions. To maintain sealed blinding code at each clinical site, firstly, to make them available promptly to address in case there is a serious adverse event. Secondly, to allow the FDA audit to confirm whether the products are actually dosed to the subject as the report to FDA. The third, to link BE reserve sample to the actual dosing. That's the answer for this question. Thank you, Nora.
Okay, thank you. And Dr. Li, will go ahead and ask you another question. Does FDA allow electronic records to be used in BA/BE studies, including IVRS and IWRS?
Thank you, Laura, for the questions. Yes. FDA allowed electronic records to be used in the BA/BE study, including RVRS, RWRS. But however, they must be secure and available for FDA to audit. Thank you for your questions, Laura.
All right, thank you for those responses. Now we'll move on to some questions for Dr. Chen again. Dr. Chen, does clinical endpoint BE studies have to comply with Part 58 Good Laboratory Practices regulations?
No, for the clinical endpoint bioequivalence studies, is not within the scope of 21 CFR Part 58 Good Laboratory Practice Regulations. Thank you.
Okay, thank you for that information. Another question for you, Dr. Chen. What are the purposes of blinding certain personnel who conduct clinical endpoint BE studies?
Thanks again, Nora. The purposes of the blinding personnel are to minimize conscious or unconscious performance bias and to maximum validity of the results for clinical endpoint B studies during execution of our clinical study. Thank you, Nora.
Thank you so much again for that response. Now we have a question for Dr. Ye. Dr. Ye, if the clinical site did not have reserve samples retained on site, but shipped to a third party, what can the clinical site do?
Since the clinical site shipped the unused and the used IPs to the third-party storage site, they can request the third party to ship it back to the clinical site when the inspection is conducting and the data is available for review and verification.
All right, thank you so much for that response. We have another question for you, Dr. Ye. If the clinical site did not have reserve samples from some shipment, what can the clinical site do?
I think that's a great question. If there's used and unused IPs from that shipment available and can be verified, which can be used to mitigate the impact of not retaining reserve samples from that specific shipment. Thank you.
Okay, thank you for that as well. Now we'll go back to some questions for Dr. Li again. Dr. Li, could you explain what is double blinding study and what is single blinding study?
Thank you, Laura, for the questions. The double-blind study is basically both subject and the site personnel are unaware the actual identity of the products. So for the single blind, the only subject are unaware of the actual identity of the product. So that is my answer. Thank you. Thank you for questions.
All right, thank you for your responses. Now, We have a question for Dr. Chen. Are informed consent and human subject protection required for clinical endpoint bioequivalence studies?
Oh, yes. I mean, clinical endpoint bioequivalence studies must incorporate informed consent and human subject protection. Institution Review Board, I mean the IRB, have the federal mandated responsibility to review clinical investigator involving human subjects to ensure that a proposed protocol meets the appropriate ethical guideline before subjects may be enrolled in the study. Thank you, Nora.
All right, thank you for that response as well. And now another question for Dr. Ye. Are retention samples still considered representative if they are shipped to a third-party storage location for long-term storage, or must they remain at the clinical site?
As long as the clinical site has a contract with the third-party storage site, as well as the third-party storage meet the requirement of the storage of that specific IP, then I don't see there's any issue for the IP to be shipped and stored at the third-party storage site.
Okay, thank you so much for that response. Now we'll go back to a question for Dr. Chen. Um, is retention of bioequivalence samples required for a clinical endpoint BE study?
Oh, yes. I mean, for the 21 CFR 32063 requires retention of the bioequivalence samples for the clinical endpoint BE studies. Thank you, Nora.
All right, thank you so much for that response as well. We have another question for you actually, Dr. Chen. What is the purpose of a placebo control group used in a clinical endpoint bioequivalence study?
Oh, that's a good question. The purpose of using placebo control is to demonstrate sensitivity in the measured endpoint. The reference product should demonstrate drug effectiveness as compared to the placebos. Thank you, Nora.
Great, thank you. I think that is all the time we have questions for now. I want to say thank you again to our presenters for all the information shared. We will go ahead now and take a quick break and resume again at 2:55 Eastern Time. Thank you.